Right. So we are a biology club. We are part of Makespace in Cambridge, and we hold these meetings on different topics every second and fourth Monday of the month. So you can join us on Meetup, and that's where we update all our schedules and descriptions of the upcoming talks. And if you wish to contribute to the Biomakespace and then Biology Club talks, you can stay up after this meeting and we can discuss about it. So today we're going to talk about lab automation, and it's an open discussion to see how different fields can help in automating small aspects in biology labs. If you're a biologist, you'd already know how we struggle with repetitive experiments or repetitive tasks in the lab, and how new introductions such as the OpenTrons robot or any liquid handling robots and programming are actually helping biologists to automate and then make these things interesting. So it'll be an open discussion. We have a mixed group of audience here. We have people from physics as background and instrumentation, and we have biochemists and biologists in here. So I hope this will be an interesting session for everybody. We have Nicole Prandi. Dr. Nicole Prandi is a research assistant in Ginny Malloy's lab. Are you here already, Nico? Awesome. I am going to stop sharing my screen, and I will. Okay, you can start sharing your screen, and you can continue.
Okay. First of all, thank you for giving me this opportunity to invite me at this space. My talk probably is gonna be, I don't know, maybe it's gonna be too technical for some and not enough technical for others, because I'm a biologist by training. So, and I just started 1 year ago after being lots of time repeating always the same experiments, I thought, especially when we want to do it in large scale, I thought about getting closer to the automation world, let's say. And so I started to, at the beginning, play around with the OpenTrons. Because we were in lockdown, and so basically the OpenTrons, I wanted to know how to use it, to learn how to use it, but we were going in lockdown. And so basically what I did was to ask if I could carry the OpenTrons, which was never used in my house. And so at a certain point in my kitchen, I had this robot, which you're gonna see in a slide later. So this is how I started. So this is, I'm gonna go in presentation mode. That's it. So basically, when we think about a robot or something, we think about something like maybe like this, or more realistically about something like this. When we go to a lab robot, what we have in front of us actually is more something like this, which is more like a cabinet in which there is some 96 well plates that some of you may be already familiar with, and then there's just one arm that goes around picking. These are the big fancy ones. There are even fancier ones which can go even higher throughput. These are like mechanical, so they have basically pipettes that aspirate the liquid. Then I'll show you a video and you're gonna be able to see it better. But there are some that the newest generation which use a different technology. For example, this is the Echo, which basically uses like sound waves to transfer very small amounts of liquid into from one sample to the other. And this allows to scale down into just like nanoliters of liquid, and at the same time it reduces the use of tips, which is plastic, so it's good, and it allows to handle a larger amount of samples in a space which is not, I put this picture because you can see, for example, this one compared to this huge one, like you can have something more compact. In our case, we have, like, I'm gonna talk mostly about the OpenTrons in this video because I was told that you have one in the space. And so hopefully this, if you never used it, it can help you to think about how to, what is the logic behind the programming in the OpenTrons. And if you have already some training in the programming, it's gonna be very easy, but also if you don't, if you're just a biologist, it's not so complicated once you get into the logic. So why automation? Why these robots? As was said before, because we don't want to spend all the day pipetting. Sometimes if you are a biologist, you sure had this problem of being there and just being like, oh no, I need to repeat this like 20 times and it's just transferring liquid. And I remember like talking with a colleague and she was saying like, oh, they think we are very smart here in the lab and what we do is actually just being lab monkeys, you know, and if this can be done by something else and we have more time to think about the experiments, that would be nice. I like pipetting, but not when it's too repetitive. And also like maybe one is tired and you're like, why this PCR didn't work? Did I put 2 times the reverse primers, for example? Did I didn't put anything in there? Is there the template? So when you have the robot doing that, you can stop doubting yourself basically about these small things. And of course, when you have a big experiment, and I'm talking here maybe more about industry, but also in the lab, when you want to try a lot of different conditions in your experiment. For example, when you want to optimize your conditions, then you want to try many different as possible, and then the robot helps. What the OpenTrons can do a lot of things. Like here, today I'm gonna talk about setting up PCR reaction. And I thought there were mainly biologists here. So if there's someone, I don't have a slide on explaining the PCR reaction, how it works, my fault. But it's just to amplify the DNA. So you have to pipette into, you have one template DNA and then 2 pieces of DNA, which anneal at the end of what you want to amplify, and you add the enzyme, and the enzyme uses these pieces of DNA to just copy. It's just copy paste, basically, the PCR. You have a piece of DNA, you want to copy paste one part and paste and amplify it and make loads. Or the mini prep, which is basically just extracting some DNA. Or for example, if you want to make your own plasmid, which means like assembling pieces of DNA, it's all about, at the end, all this application, it's all about transferring small amounts of liquid, which is something that the robot can do. And so here's a video of our robot, which is, I didn't want to, it basically makes the sound of a very angry printer. I don't know if you have those printers, that's the same kind of sound. And so what it does here, like in a couple of seconds, you pipette 4 rows, which is quite a lot if you have to think to do it them by hand. Of course, you can have a multi-channel. You can do them by hand very quickly, but yeah. Is it going? Yes, and this is basically filling one plate in a big plate in a short time, in which you don't have to think, oh, have I done this row? Have I done this one? Have I already put here? You know, it's all a matter of also reproducibility of the experiments. And here there's another video of the OpenTrons. Just it picks up. These are the tip tracks you can see here. And these are various liquid reservoirs, and here it pipettes. I think it's the same video, and this is the trash bin, so it drops the tips there in the end. Here it doesn't because it was just pipetting water. And today I'm going to talk about automating a PCR setup. I'm a bit conscious of time, so I may skip some slides, but I'm going to give you all these presentations. So if someone wants to try to, I'm not sure if you have the PCR module inside the OpenTrons, but still you can automate the PCR reaction and then put your reaction into a PCR machine outside of the robot. And so basically, to program the robot, what you need to, at the beginning, you need to ask, what do I need and where it is? Like, it's similar to when you set up your experiment in the lab. So the robot has this kind of, this is seen from the top, and it has this sort of scheme where everything is has a number, every position has a number. So you can see here there's something in position 4. This is the trash, which is fixed. This is like 2 of the modules. This is magnetic, and this is to heat up. It's like a heat block. And so at the beginning, of course, you would need the pipettes, and there are some single channel and multi-channel available. And each of those has a name, and this name is in the so this is called the OpenTrons AP language, which is basically the language in which you program it. Now they also have online, if you don't want to go into programming, they also have online a tool to just set up your protocol using a user interface. But when you need to tailor the small details, then it's useful to know this kind of things. So each of those has its own name, which you don't have to remember, you just copy paste. And then you have to specify if it's if this pipette is mounted on the left or on the right. And then you specify where is the tip rack. I showed you before in the video. So you place it wherever you want. And so you're gonna say, you're gonna make a variable which you call, for example, the tip rack, and you're gonna say that it has to load the labware called tip rack, which is, this is the name fixed, and it is in position 1. So in this moment, the robot knows that there is a tip rack in here. And then you make another variable which you call the pipette, which is this, in this case, not labware, is an instrument. And you add it on the, here you say it's mounted on the left, as I said before, with the name. And you associate here the tip rack. So you say this pipette has to pick tips in this place. And that's it. I see there are comments now. I won't stop to go to the comments. Just if I go out of time, please let me know, like verbally. So here there are the, we know that the tips, and then there is we need also, other than the pipettes, we need the plates. And all the plates, they have names, like different, for example, from these brands, they have names which are specified on the OpenTrons website in the OpenTrons library. So at each of these APE names corresponds, in the robot's mind, let's say, some kind of measurements. For example, this one is a rack which holds Falcon tubes. And it so it has a footprint which is quite standard because it needs to fit over there, but especially is important at the height, because the robot knows where it has to pick in here, which height it has to go to pick the liquid. Oops. And yeah, and so, but also, other than the things you have, if you have special, if you have some ideas, you need to do something particular, you can also design your own labware. For example, in our lab, we were trying to design a vacuum manifold to fit into the footprint of the OpenTrons. And so you can also make your custom labware modules. It doesn't have to be in the labware library. You can make your own, as long as you measure very precisely this, and you can make your own custom module. And at this point, you need, as in every experiment, you need to know the volumes you need to pick up. And so you have to define some variables, which are the volumes. And this, I find it quite useful to put them at the beginning of my protocol. So if I need to change the volumes, I just change it once without changing every time. So here there are also, other than pipettes, we can also have some instruments. In the OpenTrons, they are giving more and more instruments. This is the modules that they were available at the time we bought it. This is a magnetic module, this is a PCR machine, and this is a thermoblock. Now they are making probably, they made another one, they're probably making other 3. But here, when you put the module, you have to specify where it goes. And so you, In this case, I don't know if this is useful for you because you don't. I don't know if you have the thermocycler module, but it just sets up the profile of the thermocycler as you would do with another PCR machine. Just the number of cycles and then the profile that repeats over time. So this is the beginning of the protocol as you would see it at the beginning. So you just say, as we've seen before, if we go step by step, this is just loading thermocycler, loading this module, loading the plate in this call like this, and then the tip rack in this position. This we already seen, and then the thermocycler. And this is just the first step of the code in which you specify where things are. And where the robot needs to find everything and also the variable. Then there is another part of the code which is actually made by the action that the robot does. This I'm gonna skip. So it's divided in basic commands, and the commands are just saying the pipette what to do. For example, pick up the tip, drop the tip, or aspirate. This is the volume in microliters, and this is the position in which it has to aspirate. So, for example, here it says it goes back to this variable we defined before. So it knows already that it is in position 2 because at the beginning we specified that this plate we call like this, it's placed here. And for example, there are different ways to do it. Here there is, you can say put 11 microliters, or you can put another variable. You can say, if you defined before that the water volume is 11, you can say just aspirate the water volume. And then this is aspirate, and then there is dispense, of course, because you pick up the volume and then you dispense the volume. And here you specify where you dispense the volume again. So it picks up from this plate and it drops it in the plate where we want to do the PCR. And as again, we define this variable. Then there are also some complex commands such as transfer. And transfer includes to pick up and to drop. So to pick up the tip, to aspirate and to drop. So we have the water volume, for example, here, that we want to take from here to here. And there are different complex commands, transfer from one well to the other, or distribute, aspirate from here and then dispense, or the contrary. And this is like an example of the pipette, which is aspirating from here and distributing into this plate. As you see, it picks up once and it distributes. And this is saving you time, especially when you have this kind of plates with a lot of wells. And here I conclude. Oh, here I conclude because I was talking about the PCR, but for example, what we try to do is also to automate a mini prep. And if someone of you used some mini prep kit, you would recognize this kind of pink liquid that can be pink, can be blue from kits, from mini prep kits. And so here we were, in this case, it's mixing. So it goes up and down as you would pipette into the well, then you can ask the robot to do it instead. It's quite useful when, and this is the alternative of, for example, of a vortex that we don't have in the robot. And so this is a script. Maybe if there's someone from you in programming is gonna laugh at my script because it's very elementary. So it's just saying in the end to transfer the liquid, all the liquid transfers one by one. Now I probably able to do this a bit better, but, and then it says, it uses this command transfer, and then it says to mix after. So it's just how you would pipette, put in another place and transfer. And yeah, this I think I can skip, and I can thank you with this image of the OpenTrons in my kitchen table, which would make a nice interior decor element. I'm missing it a little bit now. Thank you for listening.
Thank you, Nico, there. Thank you very much. We can take some questions now if anybody has some questions or somebody wants to kick off the discussion.
Hi, I have a small question. Does anybody in the group try to make OpenTrons hardware? Because it says open, but does anybody try to make robots similar to OpenTrons? Maybe Nico, did you ever come across anybody trying to do this?
To make hardware for the OpenTrons.
Yeah, building from scratch.
Yes, we were trying. We were in collaboration with some engineers because we don't really have the tools. We were trying to make a vacuum manifold from the for the OpenTrons. So it's a whole new module which just needs to create some vacuum to use, because like now to extract DNA you have to use magnetic beads, but we wanted to try to use the column because we were trying to automate the some COVID testing that they are doing in Ethiopia because they got an OpenTrons. So we were trying to automate their existing method, and we have the vacuum manifold there. Is still, unfortunately, is still missing the part which interfaces directly with the OpenTrons. So the part that says on and off, just going through the OpenTrons instead of having to do it from outside. And this was, yeah, it's still we're still working on it.
Okay, not just about one module. Does anybody try to do entire OpenTrons again from scratch?
So you mean building OpenTrons from the scratch, like?
Yes, like a 3D printer. It's based on a 3D printer.
Yeah.
When they made it, initially that was 3,000 euros or something, I think. But then now it's quite expensive. I mean, at least double this price. But
Yeah.
I know a few efforts people have been trying, Pipette Jockey and all. But he tried to contact them, but really at one point it is not possible to use their software API to communicate. So I would like to know if others have tried this and if they would like to share that thought, or if anybody would like to make OpenTrons, what would be the most critical part?
Yeah, I don't know about the hardware, but I could say the software is one of the key things. I'm a biologist myself. So 2 years back, we had a point where we need to discuss in the lab to buy an automating robot. And we were kind of puzzled between the Gilson robot and the OpenTrons and also the Andrew Alliance. So what made the discussion in the favor of Andrew Alliance was the software. At that time, OpenTrons was still a bit of coding. They had support from the communities online to give protocols, but the Andrew Alliance had a drag and drop interface, and that basically meant a big deal for all the biologists. They didn't want to code. It was precisely, they had these icons of little pipettes and little tubes and plates. So all you need to do is just drag and drop, and then it'll ask you how much volume do you want a pipette, and you get it in there. And the protocols were there made in a library as well. So you all need to do is just drag and drop. But the difference is the OpenTrons was about $3,000 at that stage. Now the Andrew Alliance for us costed about 37 grand. But that didn't stop there. All these racks for the pipettes and the magnetic ones, they were called dominoes in Andrew Alliance terminology. And each of them costed about 500 euro just for that 3D printed. And I remember China actually 3D printed a few of them, but somehow they weren't compatible because the camera wasn't detecting the proper codes or something. But from since then, the OpenTrons have come a long way.
Yeah, I think now they have basically most of the functions, they are in the online in their online software. So if you don't want to program, you can still make a nice protocol out of it. I wanted to program. I'm a rare case of biologists who wanted to program, but I realized that it's not very.
Yeah.
I would add the Andrew Alliance is a lot more sophisticated hardware. I think between the camera that they have incorporated in there, and then even things like the motors themselves are much better. It handles failure a lot better, but it does cost 10 times as much. As far as software, I think the my experience with the Andrew Alliance, and they told me that they were going to keep working on this, was that it was harder to program. You could do simple things pretty well, but if you wanted to make a template for a new kind of procedure, it was much easier to do on the OpenTrons because you could do that kind of programming. And then just Chinna, to your point about building it, I haven't built one from scratch, but we spent a long time working on an old OT1. And gosh, it's a lot of work. I would easily, if I had the budget for the 3,000 or 4,000 euro, I would much rather do that than spend that time building stuff, because there's just a lot of moving parts. But if you want to try, the smoothie board is really the core of it, because that drives the motors. And then there's a lot of work on the software side, which you can just take OpenTrons' version of that if you can get close enough.
Oh, thanks, Tim, sharing your experience. So on the smoothie board side, do you think OT1 is completely from the off-the-shelf smoothie board, or is it a variant of the smoothie board they developed?
The newer version, I think
No,
it
no,
is a
not the
variant.
newer version, the OT1.
Yeah, the OT1, I think it's pretty close to off the shelf. I think you do need to flash new firmware onto it, but that's provided in the OpenTrons repository. It's on their GitHub. It's not too hard to do.
Okay, and you did that by yourself?
Yeah, we tried a lot of versions of it, and in the end we gave up. It was just someone was willing to donate us an OT2, and it got us faster to what we wanted to do.
Okay, I hear that the most difficult part is the pipette. Using electronic pipette, hacked electronic pipette is solve some of the problems. What is your take on it?
Yeah, we never got very high precision, but also we just didn't. The things that got in our way, we had bigger problems to solve. But yeah, I could tell that the very high precision on the pipette would have been a challenge, because you're trying to control volume by how much you move the robot's version of the thumb on a pipette, which is not a very precise way of pipetting. So yeah, so.
Great, thank you. I might ask you more questions, but let's see what others are.
Just a comment on the OpenTrons, because it's so cheap, it's about $3,000. As a biologist, do you think if we fix an assay for each OpenTron, say about you're a microbiology lab or a genetics lab, you have DNA extractions, and DNA extraction pretty much as a day-to-day kind of work. So if you can fix that $3,000 instrument just for a DNA extraction, but make some modification around it so that you just open the DNA extraction kit, put it aside, and it does the rest until the end. Not just the robot, but having small modules around it that quantifies and then gives it back. That'll be very interesting. I've come across this digital to biological converter that was published a few years ago now. It's in Nature. I'll put the paper in. It's actually quite cool. They actually made the whole automation for biologics production. It's all automated. I'll drop that link in the. It's by Hunson Robotics.
Nico, I have a question. Do you prefer the sort of, now that OpenTrons has the graphical interface on the web, but you've had the experience with the programming, do you still prefer to do the programming, or is the graphical just easier for you?
No, actually, I'm still sticking with the programming, because we're doing new protocols and maybe unusual stuff, and it's and I need to have the most control as possible on it. For example, we were doing something on the something with magnetic beads that would settle down the well very fast, very quickly. So we had to increase the speed of the pipetting out to make them move and to mix them, because if not, they would just don't move. And I think with the graphic, you can still control the speed in which you pipette out, for example. So there are a lot of small things that you can tweak with the programming so this is why I would stick with the programming still. Maybe if when I got it, everything, all the functions were able on the graphic interface, I would have gotten that. But now I'm sticking with it because I can see it better on the, how it goes.
That makes sense.
Hi, Nicole. So I put my question in the chat. So about the DNA RNA extractions. So which kit do you use?
So for the DNA extraction, actually, I was starting to automate one kit by New England Biolabs, but those uses column. And so what we did later was to so the thing we are using now is not a kit, is some buffers we made. So there is this, it's no more based on column, it's based on magnetic beads, which is the optimal system to extract them. But the kits are quite expensive. And so we optimize our own method in which, so if you go on a website, it's called Bomb Bio. And they have an open, they put all their methods and protocols open source, and they have these recipes for the buffers. So we started from that recipes, and we are using those at the moment. We're using homemade buffers, which makes it much cheaper to do the DNA extraction. RNA extraction, I have to try to automate it still. I haven't tried yet though. So I can't tell. I know that it's possible to do that because the OpenTrons, the company set up some COVID testing they set up this COVID testing platform made out of 4 OpenTron robots, one in a series, and they were definitely doing the RNA extraction. So it is possible. It's possible. We haven't done it yet in our lab.
So it will be mRNA enrichment, I assume.
I think it was just an extraction kit. For example, the thing they were using in Ethiopia for the COVID testing, it was just an extraction kit, and then this RNA was going into the PCR later, into the amplification.
Okay, thank you.
I'm not. I see your other question, but I'm not sure about this application. Yeah.
It's, I thought maybe someone tried single cell RNA-seq. Anything, because that's why my question was what the minimum volume that you can use and how accurate is it?
It's one microliter, and so it could be better. As I showed you before, there's another platform which does really tiny amounts, and that is probably way more precise. I haven't done it. I would be curious to see how accurate it is. For example, if I want to do some quantification, but they use it for this kind of application, which then you have like a RNA amplification, and you have a threshold that you need to reach to have a positive. So it's good enough to do that, definitely.
Okay, thank you.
If I can answer another question, which I can see in chat, because I was typing, but then some people would ask me. So there was a question about how do you design new supports? So we used CAD software for 3D design, And we designed them so that, and also the question says, how do you tell that the machine specification, or you tell the machine the specification of the custom support? So basically, of course, we can do only the some particular measurements in terms of footprint. It's fairly standard, as I said, because it needs to fit over there, and the height cannot go over a certain amount. Then we designed, having this restriction, let's say, we designed them using a 3D software, and we 3D printed them. In the case of the vacuum manifold, it was still following this kind of measurement, but was made of metal instead, for obvious reason, because it has to hold vacuum. And then there is, to tell the specifications, basically, this is quite straightforward to do. On the website of the OpenTrons, they have the custom module thing, and they just show you a form, let's say, that you have to fill in with the dimension of your new rack or whatever. Then the spacing between each well, this is one thing that it has to be regular in terms of spacing of all the wells. And you specify your measurements over there, and then it generates a file which you then input into the OpenTrons, and so that the OpenTrons has a new definition, so that when you put that name in, it calls your custom definitions. But it's something that they just give you the file to import in your OpenTrons. It's quite easy.
If there are no more questions, I would like to ask again about the hardware of building the OTT. I'm very interested in that. So now the 3D printers are at least 10 times less in cost, for example, Creality kind of things, of the similar build volume like OpenTrons. So if anybody, because it's a Makespace, meaning that many people are already equipped with the skills to build such kind of equipment. Is there anybody interested to build such a device?
I can't help to build, but I can test it if you make it. I can see if it works.
Okay, that's great. Thank you.
Well, for those on the call who are in Cambridge, if you want to fix an OpenTrons one, then that would probably be the priority, because we have one, but it hasn't been, it belongs to the Synthetic Biology Society at the university, but they sort of brought it to Biomakespace for general hacking purposes. It's currently sitting in Biomakespace, but it will of course move to Makespace, as Biomakespace will do. So yeah, there will be an OpenTrons that is fully assembled, but just, I mean, no one's used it for some time. It was a super early model as well, so there was some issues with the pipettes being ejected and other things, which were fixed by a custom 3D printed edition, and then I think they upgraded the pipettes later. So it should, in principle, it should work. It was working. It was packed into a box. It was unpacked from a box, and then it's not been touched since. But yeah, if people want to start with some fixing / reverse engineering, then you'd be very welcome, I believe, to have a look at that. I'm still waiting to hear from the Synthetic Biology Society if they have any specific plans for it, but they didn't last time we spoke.
Okay. Great. And also the drag and drop software, I looked into their drag and drop software. It looks good, but while looking at the codes and commands what Nico showed today, is there any Scratch type of language developed for OpenTrons? That would be, a little bit better for the biologist.
I don't think so.
Okay. And do you think anybody who uses of OpenTrons that such a software would be useful, like Scratch?
So I'm actually working on a project kind of in this general area to sort of figure out a good way to design experiments that kind of is in between the sort of drag and drop. It's a really hard problem though. If you know, if you can learn a little Python, that's, you know, a pretty fast way to get up to speed doing more complex experiments. And then the drag and drop, I think, is pretty good for very simple things or standard things. But yeah, I think a Scratch-like interface would be really helpful, and that's something I've been working on to try to figure out.
Okay, thank you. What I mean, maybe I need to explain here. Scratch is like, it's like small building blocks like Lego, but it's like pre-built commands that you can drop and drag, and they click to each other only if they're compatible. So there is less chance of making mistakes, particularly you know, people who doesn't have great experience in programming. On the other hand, these are, as Tim explained, not easy sometimes to build, but if they can be, I saw some people working on such kind of languages, Tim, particularly using the Google's what we call in the app inventor, they use this code blocks. I forgotten that name now. Blockly, I think Blockly is the one. It's not just for the liquid moving robots, but many other biology experiments. If you have that kind of interface, it automatically generates the protocol, and it can be useful to be in a notebook or something. I think already OpenTrons generates the protocol for you to have it, revise it, and see it. But you know, such kind of drop and drag and drop will be useful for the biologist. I don't know how useful their graphical user interface based on this blocks of diagrams, but Scratch kind of thing has more support from because they are developed for children. There are many companies now using the same programming structure to teach children coding. So biologists in a way are very close to children when teaching the programming to them. So I think that would be great.
Although there's an argument for upping our game, being a bit more aspirational when it comes to learning programming as biologists. I mean, I think things are slowly changing. I mean, it is becoming more and more necessary, whether you're heading into bioinformatics or whether you're kind of basically doing anything in molecular biology, to have a little bit of programming knowledge. So it might be that the kind of people that are coming through the biology degrees now will end up learning a bit more.
I mean, I come from the opposite side, where I kind of started with the programming and then worked the other way. So you won't hear an argument from me. I think learning a little programming is super helpful.
Yeah, absolutely. I mean, it's not so automating stuff in the hardware sense is useful, but also automating stuff just to like download DNA sequences and kind of but you need an application, which is why automating stuff in the lab to save you time is quite a compelling reason for someone to learn a little bit of coding. I didn't really learn. I mean, I took a couple of courses, but I don't think I really learned. And I mean, now I've forgotten it all, but I learned a bit of Python just because I had to download thousands and thousands of DNA sequences, and there was no way that I was doing it kind of manually. So it's a good motivation if you know you'll eventually save yourself a bit of time.
Yeah, one thing to add in there, again, coming back to the Andrew Allen's robot in there, I've used the software. I really like that software just because it's drag and drop first, but then as soon as you say, okay, this is the experiment I'm going to run, it's going to generate a protocol for you and then run through it in a brief kind of summary to check for all the parameters, such as changing the tip in between or checking if the volumes are correct. And I don't know if we have that flexibility, if we write our own programs, we need to have another program to check what we've written, like just like the simple things, changing the tip after every mix or something like that. Or I think there should be a nice skin or a wrapper, software wrapper, to go before and after the custom programming for every experiment when a biologist writes a code.
You know, talking about the OpenTrons, it runs a simulation quietly before it actually does anything. If you upload to the OpenTrons app, you may get an error if you wrote your code wrong or something. And I could imagine adding kind of some of the features you're talking about.
Is it the latest version is doing that? Because when this is about 3 years ago or something, I didn't have that facility at that time.
Yeah, I don't know when it started. Maybe in the past few years. But you can actually test your own protocols without a robot. You have to download their software, but it's called OpenTrons Simulate, and it figures out what the robot will do in each step, but it does not do the thing that the Andrew does, which is figure out all the consumables you'll need. And
yeah. No, even the notification systems when something breaks, the camera taking pictures at every time to have a back end to troubleshoot. I think they just had a little bit more money spent on the software than the OpenTrons guys at that time, but I think OpenTrons is picking up right now.
Yeah, totally.
Hey, Tim, long time no see. I don't even remember me, but I used to hang around like CCL a lot.
Yeah,
But hey, how's it going? Yeah. But yeah, are you still doing, I got to this meeting a little bit late, just because I was in another meeting for work before this, but are you still doing the color plates with the OpenTrons at CCL?
Yeah.
Nice.
I mean, I guess a small plug for this group. I don't want to take up all the air, but yeah, a lot of this OpenTron stuff that I've my experience with it has been hacking it relentlessly for this BioArt project called BioArtBot, about making the OpenTrons kind of draw BioArt as kind of a learning method and trying to figure out what is the sort of easiest way. So yeah, we're still working on it. We're trying to figure out exactly what the next steps are right now, actually.
Nice. Yeah. Glad to see you're still working on that.
Yeah, thanks for asking.
Yeah.
Yeah, if anyone wants to check it out, it's bioartbot.org.
That's fun. We've been trying to entice some more artistic folks along to Biomakespace. We had a agar art night, but we haven't done anything involving robotics yet. I asked a question in the chat if anyone's automated stuff other than pipetting or would like to, because yeah, in general, most labs, unless they have a very large budget, have automated almost nothing in the biological space. And I think there's a lot more that you can do beyond moving liquid, although that is like up to 90% of what a molecular biologist does on a daily basis, so it's important. You said you've been doing some bioreactor automation, Hans.
Oh yeah, yeah. So I worked on this for a long time before coming back here to Georgia. But I got just a Raspberry Pi, and I knew Python, so I just wrote in that code. But I automated I guess you can call it a bioreactor. I don't know. It's just a mason jar, and it has some tubes going in and out of it, and everything's connected with like little autoclavable lure locks and stuff. But yeah, you can grow bacteria in it. You can just, I put usually like LB media in it, and I would grow E. coli in it, and then it would regulate the temperature of it, because I had a system hooked up to the Raspberry Pi, just like a little heating pad around the mason jar. And and then a thermo probe just kind of stuck into the mason jar. And I made sure the thermo probe was autoclavable too, so you just sterilize the entire thing at once. So it would keep the temperature, and then it would also constantly blow in air to it, and just to aerate it. And then it would and then my latest project on it was to try to get optical density readings on it, so like OD 600 nanometers, so you can see kind of the concentration of the bacteria that's growing. That did not go well. It was weird, because I got like I tried all these different ways of like getting an orange LED and a photo diode on the other side, and like running the culture through a tube and stuff and trying to like detect that. It turns out I could measure the temperature of the solution much better than the actual optical density of it. So like as it went up in temperature, the optical density reading went up and up as well. And so after like months of trying on that, I finally figured that out, and I ended up using a just a standard like turbidity sensor that you like put into dishwashers. That actually worked best. And then it just like runs the solution through that, and it measures the turbidity based on that. Yeah, and it works and everything. I just never got around to actually putting it online on like GitHub or anything like that. And then I got a job at another big corporation, and now I probably can't put it online because they probably come and like try to sue me or something. So, but yeah, it's possible though. You can automate almost everything. I mean, Python combined with Raspberry Pi is just amazing. And you can control motors so easily through it. I mean, they have all the systems set up and everything. So it's pretty, I wouldn't say easy. There's always a lot to learn on it, but it's pretty cool. And yeah.
Nice. Has anyone in the room got something that they're like, I really hate doing this in the lab, please automate it for me? I don't know if you've already asked this question because I was late due to losing my phone while walking the dogs. I had to repeat walk the dogs in order to find the phone. So sorry if I'm repeating a question, but anyone else got anyone who does biology stuff and wants to free themselves? Washing, yeah. Well, we actually the OpenTrons that Biomakespace has was bought originally for the plant sciences department. The only reason the Synthetic Biology Society ended up with it is because the plant sciences department lab upgraded to an OT2, so they didn't need the OT1 anymore. But yeah, they basically used it for well, subculturing, just anything that involves like washing or splitting cultures of cells is quite nice if you can get someone to do it. Although I guess washing E. coli cells is a bit more difficult because you've got to include centrifugation unless you've got some membrane-based system. And like Nico said, that the problem with some of the robots is that you've got to interface with other machines, and that gets super expensive. Like so there's a machine, and maybe Nico, you already mentioned, I don't know if you like mentioned other automated platforms in your talk, but there's a machine called Echo, LabSite Echo.
Yes,
you did mention that. Okay, cool. So yeah, the LabSite Echo is super expensive. I think it's like 250 to 300,000 for the base unit. And they do sell, it's basically like an arm that loads the plates into the device and takes them back out again. But that module is tens and tens and tens of thousands of pounds just to have the plates loaded in and out. So most people don't go for it. But like it would be really cool to be able to have things unloaded and loaded into centrifuges. That would be very helpful. That's pretty much where I think, Nico, that's kind of the bit where most of the parts we end up not being able to do is just because we have to centrifuge it.
Yes, exactly. Although like there are some parts, for example, like the mixing, the vortexing and the incubation. And basically I wanted to tell you, maybe I didn't, but basically I was contacted by the OpenTrons that they are planning to do not a centrifuge module, but a vortexing module and also a heater shaker, which would be very interesting. Because it, for example, if you want to do a transformation, it can have even the shaking between that. And so they are progressing through that. But yeah, yes, the centrifuge is the bottleneck. And like even when I was in Manchester, they would still have a centrifuge outside the robot, and you need to spin down. They were trying all the possibilities to avoid the centrifugation step. But sometimes it was still needed some person to go there and load the centrifuge. We were trying to do the vacuum manifold, but to at least to have we actually use that to both to purify DNA and to purify proteins, and it worked, but yeah, I guess there is some optimization we need still to do, because that having the vacuum, it could for some application is not in those where you need an actual pellet, but other ones you can substitute a centrifuge.
So one quick question, just out of curiosity, because you mentioned those models. Are these modules built like for the whole plate reader, or do the robot moves the liquid into a separate I don't know, like separate a container to move them into the separate module?
No, I think because this robot doesn't go outside. There are some robots or some things that they have an arm that picks a plate and brings it outside, but this is all in that box. It cannot go outside, so the module has to fit inside of that square space.
But do you mean like the module would take the whole plate reader and centrifuge it?
Sorry?
Would this module take a plate reader loaded with all fully loaded with liquid and centrifuge it?
No. This is just a shaker that they were thinking to build for the OpenTrons. They're still developing, so I don't have details, but.
All right.
The dream module would do that though. The dream module would take the thing and centrifuge it, like take it off. And some people have put arms outside the OpenTrons that will move plates from one OpenTrons to another. Like they've kind of, changed them up. But it's getting it out of the OpenTrons into a centrifuge, switching the centrifuge on, because centrifuges are actually quite tricky to build is the other thing. Like a lot of people have kind of automated stuff because they've hacked something together. And if you're not doing anything too mechanically strenuous, that is okay. But yeah, hacking a centrifuge is a little dodgy, because you really, really, really need to know that whatever you've put to control the lid, for example, is not going to fail mid-run and have the centrifuge lid open, because that would just not be safe. So, I think that's a bit of a concern. I haven't seen that many people messing around with centrifuges.
Yeah, there was this paper I didn't understand really, because they were talking as the centrifuge was inside the robot. It was one of the big fancy robots. But I couldn't find any more information about it. If there is actually a tiny centrifuge inside the robot, that would be strange. But
I'm
they
sure
were
some of them must have one. I'm sure some of them will have a centrifuge that they've managed to squeeze in there.
Yeah.
Interesting enough that people are trying different models of automating things. So BGI, which is a big DNA sequencing company, their latest sequencer, they kind of used to have one that you basically, everything was a bit more contained, like it looked like one solid instrument, but at a certain stage things had to be kind of taken out and put into another instrument. And I visited, this was 2018, I was at the China National Gene Bank and sort of doing a tour around, and they had prototypes of their latest version, which is basically like a robotic arm that's in the middle of a set of instruments. And so the robotic arm just kind of moves the plates out and into the next instrument. And so it looks a lot like, in a sense, it looks a lot less sleek than the previous models, but apparently it's way more efficient to have it done that way than before, when they were basically trying to kind of I don't know how they were channeling the plate through, but they had more manual steps, and they also had more sort of, it would pass through an instrument, if that makes sense, rather than being taken out and moved around. But it was quite cool to see it in action, this sort of robot arm flipping around, a bit like the ones you see on car adverts that can move in any direction and kind of pull things out. I'll see if I can find a photograph of it and post it on chat.
I have a question for the group that's a bit of a pivot, but I think related, which is how, I guess I just want to get a sense for how much people know about collaboration tools like GitHub or things like that, and how interested people are in it, because it can be hard to find who is doing what, and if someone has already solved the problem, I don't know. I worry sometimes that we're not actually collaborating enough, doing these kind of DIY bio stuff.
I think that's a great question. I barely know anything about collaboration tools. I think I tried looking up things on GitHub and stuff, and I, for one, I would love to know more about GitHub and everything, but the time and stuff to learn that, it seems like will be daunting.
I search extensively in GitHub, Thingiverse, YouTube, on anything which I would like to build. First of all, sometimes building itself is a hobby for me, and it's like a play. So I like it, even though somebody built it. Maybe I might make some small changes, and I like that way. But collaborating, I try to, but in sometimes it depends. If it is not a serious research project, people reply at different times. Even if I ask question in GitHub, people don't, the main authors of the GitHub repository, they don't usually reply within first month or so. Then that question becomes obsolete, so the collaboration dies there. So I use these tools mostly to check what others have done, how they solved a particular problem. But directly interacting with people, hasn't been that much successful.
Yeah, I think it can be really limiting because it does take a certain amount of time. And if this was just a project you sort of did for fun, it can be hard to keep up with. Suddenly you have a new job where you have to talk to a lot of people about your project. But I guess that strikes me as maybe this is a place, for focus, is to think about how to make those kinds of collaboration tools a little easier.
Yeah, I agree. On that point, Hans, on the optical density, I tried loads of optical density instruments. Maybe one of the things you might have failed with the jar project is you might have used LED and stuff, laser and LED light might be dispersing a lot, and maybe external light influencing your readings. So a laser will be less dispersing, especially if you use a red laser. And if it is straight, the photodiode is straight across the laser. And I have seen papers which use turbidity meter, and that used in the washing machine that you can buy on AliExpress or eBay, but that's not so reliable than some photodiodes with a very linear response. So it might be taking the right photodiode and laser combination to work with large amount of fluids. LED works particularly well for small test tubes or falcon tubes, such kind of stuff. But if it's a large jar, the light usually has it doesn't propagate in a collimated beam, like a collimated beam from straight out of the LED. So that might be one reason.
That sounds like maybe what happened. So when I tried this, I was just using literally just an LED that I got off Amazon. That was like an orange LED. That's the closest I could get to 600 nanometers, right? And so, and now on the other side was like a photoresistor, and then I also tried it with the photodiode as well. And what would happen was that as it's supposed to be like as the turbidity got higher, I would get less and less signal from the photoresistor or the photodiode, right? Because I was losing light as the bacteria got more turbid. But weirdly, what I would see was like almost like I forget if it was like a U shape or like the opposite, like an upside down U shape or something, where it didn't quite go linearly, and then it seemed to have like this weird, it didn't make any sense. Like as it got higher in turbidity, it was seemed to get like lower or something when it wasn't supposed to be. But you're saying so a laser diode would actually help me maybe there, you think, or?
Basically, laser helps because you don't lose so much of light, unlike LED. Particularly the laser shape, the jar you are using, that is one, because that's a large volume, right? So as your volume increased, bacteria concentration increases, the scattering increases. So at higher concentrations, you will have this problem even higher. So best is to use a cheap laser diode that, they cost less than $1 or something, and that should fix the problem. Another way of doing it is that rather than putting the photodiode and laser at opposite directions, you can put them like side by side, like how heart rate sensors in the fitness trackers work. So actually, we did a paper recently. We hacked a fitness sensor to make it as an optical density meter. That worked very well. So so it is easy that way. You'll not miss the. You'll not be missing so much of optical density readings at higher concentrations, particularly.
Okay, I mean, can I maybe get that paper from you or something like that? I'd love to, because
I can send. I think Vamsi has kept that link here.
Oh, okay, awesome. Thank you.
Although we are using LED here, keep in mind, because our sample volume was in a Falcon tube. But if you're using a big jar, I suggest you to use a laser diode.
Oh no, this was still like, this was only in this was going through a silicone tube. So I was trying to measure it so that I didn't have to have the actual sensors in the solution. So I was just trying to put a silicone tube between the photodiode or the photoresistor and the LED.
Yeah, that's a big problem because the curvature is so steep,
Yeah.
The optical sensors should ideally work in planes, between right angle planes, not like curvatures. The curvature introduce another problem. Lens is called. So that's another problem. So yeah, have a look in the paper which we sent and the references therein. So maybe you will find the answer. There are many people who try to do these optical density meters. I read a lot and try to find out the pitfalls in others' designs to come up with my own design. Yeah.
Okay, nice. Very nice. Thank you. Yeah, I might email you later on if that's okay with you.
Yeah, sure.
Cool.
Here, since Jenny asked this question about automation, although it might not be that much of an automation, but the fitness tracker device which we built, we try to commercialize because one of the problem in biology is like taking optical density every few minutes. Many biologists spend so much of time in a day while making this growth curve. So the device which we made is that the optical density is relayed to the mobile phone every few seconds. So plot is automatically made. So you don't need to make the growth curve, in a traditional way, taking the fluid out of the Falcon tubes or whatever the cell culture. So that way we are automating optical density measurements. So that might save the time of the biologist. We got a grant to commercialize this device. So we are in the middle of that.
Nice.
One other project that came up in previous meetings, so we used to do a monthly meeting called Science Makers at Makespace, and one of the projects that a biologist there wanted was, again, washing. We do a lot of washing. They wanted to wash basically plates that they'd done. They were doing something to do with tissue culture, and they needed to basically blot some plates and then wash the Western blots. So they'd done, no, not Western blots, what am I talking about? It was protein staining, and they needed to wash a lot of plates. There was another one that was around Western blots and how to, maybe it was soaking gels. They wanted to like have a bunch of gels that they were automating the soaking and shaking. But yeah, there's a load of things like this that just take a remarkable amount of time to do, and yet, should be relatively simple to automate. But then there's always challenges when you start doing it and finding like what's the most flexible. Someone's got to design the system.
Yeah, especially for Western blots, I totally agree. It was one of the most awful experiences in wet lab, sitting in the late evenings and filling up gels and waiting for them to run. And yeah, if you have a fast transfer system, that's fine. Otherwise, waiting for the next day until the transfer happens and then staining. And yeah, no, it's one of the things I hated the most in my lab experiences.
Yeah, there's a lot of that sort of stuff. We don't have there may be an automation need for this eventually, but we're trying to shave time off of growing up cells for producing proteins by basically not doing liquid culture and just growing them on plates, which is working out pretty well actually. We get basically comparable biomass on 3 largish Petri dishes compared to a 2-litre culture. And basically scrape them off the plates the day after. We use auto-induction media, so you don't need to kind of add chemicals to induce the expression, and you don't need a centrifuge, you just scrape the biomass into the tube and off you go. Obviously you have to then centrifuge for washing, but it removes at least that step. But I've been thinking like how to scale that up in the sort of sense of, you know, just having something that basically periodically scrapes and reseeds the plates until the agar is depleted of nutrients and we have to replace it. Kind of like you know, those things that skim over ice rinks, kind of push all the stuff off.
Oh, that's very cool. So it's just like a lawn of whatever the microbe you're trying to grow is?
Yeah, it's just E. coli. We're doing what normally we're using auto-induction media that you would normally use in flasks, but we're just using auto-induction agar, and it works really nicely. They're very happy growing. I mean, E. coli aren't meant to grow in liquid culture. They're quite happy being on surfaces. So we're getting basically the same amount of protein as we would in a liquid culture, and it's way easier. You just plate it, leave it overnight, come back, scrape the stuff off, you're good to go.
That's interesting. Huh.
Yeah. I think that would actually be really doable in an OpenTrons environment. You just need to design a squeegee or something.
Yeah, exactly. We need to sell squeegee.
Should get one of those tongue scraper things or something to just scrape it off.
Yeah. Well, you can get nice disposable cell scrapers for tissue culture, but they are a bit narrow. They're mostly just for flasks, so they're quite short. But yeah, as I say, we're still testing it out at small scale at the moment to see. We haven't found a protein yet that it doesn't work for, but I'm sure there'll be some.
Nice.
Yeah, that's fabulous.
Yeah, we haven't decided how big we can go yet. I've got some students, we're trying to get some testing done over the summer so we can hopefully put out a paper at the end of the summer, because we think it will save a lot of people a lot of time. It's super easy for training as well. That was kind of part of the motivation, was we run courses, and it just saves a lot of faff. So simple to do. But yeah, if anyone else wants to try it out, do let us know. We can send you the protocol.
I would like to try it out.
Awesome.
Well, let me send you my email in chat.
Yeah, I can put mine in chat also if you
there you
prefer.
go.
Drop me notes.
Would it be helpful to increase surface area at all? Like if, you know, instead of a flat plate, you could do, you know, ridges or something? Is that an interesting path? Yeah,
potentially. It'd just be more difficult to scrape off. I mean, we're just planning to use massive Petri dishes at the moment, but not actually Petri dishes, because we're also all about not generating too much plastic waste. So we're trying basically like Pyrex dishes that you use for cooking stuff, because they're quite large and you can reuse them.
You can get like a casserole of cool.
Yeah, exactly, like a flat casserole dish.
Huh. That'd be easy to just stick in the oven to sterilize as well.
Yeah, they should be autoclavable. The thing I've realized in my preliminary experiments is that the lids that they sell with them are definitely not autoclavable. So, but they sell like stretchy silicone coverings, which are. So I think we can probably work out a way around it. Yeah, it's not automation, but it is time saving.
I know there's I did this when I was trying to design that bioreactor that I was talking about, but there's a 3D printed filament that you can get that you can actually sterilize in the oven. Yeah, the only problem is after about like 3 times, or more not 3 times, more like 10 times of sterilization, it starts to fall apart though. But yeah, if you're interested in it, it's just what is it called? It's called Spaghetti 3D printing or something, I think, and it's called like yeah, HTPLA. So.
Oh, cool.
Yeah.
I guess we're towards the end of the event. Has anyone, like, did anyone come in with ideas that they haven't said yet? Kieran, you've been quiet. Any thoughts?
Can you hear me, by the way? Just double checking. Brilliant. So I don't know. There's not anything that's really fully formed in my mind at the moment. I've got a few kind of ideas of some stuff that's been said, but I think I've just got to go away and kind of, you know, like have it in the back of my mind for a bit and think things through to kind of really contribute much to what's being said. It's in the scraping off E. coli thing's definitely interesting. The one thing I sort of wonder about that is if you wanted, there are shapes where you can, you know, have lots of folds and then sort of stretch it out, but I don't know how that would affect the E. coli colonies on top, if you know what I mean. And kind of making the depending on what the thing underneath is made of, if you're going for something like Pyrex, it's obviously not an ideal kind of thing, whereas a plastic sort of ridged thing, I guess you could remove the agar potentially and kind of stretch it out afterwards into a flat sheet. That might be adding a lot of complication though.
Well, we were thinking about other material options, like there's this thing called mirror cloth, which is kind of a lab cloth. But yeah, we haven't really tested it, but it's getting the stuff out. The mirror cloth, it's scraping it off. We basically scrape it off with a razor blade, which is quite easy. But scraping it, getting it out of anything that's sort of soggy was a bit of a problem. So yeah, I don't know, but it's worth thinking about. Yeah, it's a shame. We'll have to set up some. I know not everybody on the call is geographically located in Cambridge, but at some point when we can, we'll have to do a robotics lab meeting with the robot in the room for some inspiration. I think that's always helpful. And also one of the upcoming biology clubs, and Vamsi, it's the 14th of June, right? The Build It for Biomakespace.
I gotta check that real quickly.
So one of the upcoming, I think it's the next one, which is the second Monday of June, the 14th. It's the event. The theme of the event is basically building stuff for Biomakespace. So some ideas of stuff that we could build here, and maybe lab robots will come up if you've filtered it through your brain by then, Kieran, and you've got some ideas. Then do come back. Currently, we're going to talk about shaking incubators and oh man, I've forgotten what the other one was. Oh, transilluminators, because they're reasonably easy projects to start with, and the Biomakespace shaking incubator is broken, so that's a little bit more of a of an urgency. It sort of works, but it's a bit of an amalgam. We basically took the shaker out of the incubator and put another shaker in, but it's not ideal.
Yeah, it is the 14th of June is the next event, yeah.
Nice.
Jenny, any standard plans for the shaking incubator you're making? Any GitHub link?
Yeah, well, there's a few. I can link you to one that would be on the table as an option, but I think it can be improved. I'm
working on orbital shaker, which is not same as incubator, but you know, the orbital shaker is kind of similar. So I'm looking at different designs recently. So yeah.
Oh, cool. Oh yeah, I mean, it's basically an orbital shaker in an incubator, so it's not that's
yeah
just combine the two. But yeah, I think so. I don't know if this is public. I have some it's on a Trello board, and I don't know if the Trello board is public because it's not the Biomakespace one. I'd have to check. But I did do quite a bit of looking for incubator designs, because the nicest incubator designs I could find were actually for 3D printer enclosures. There's some really nice ones out there with kind of nice, well-designed doors and everything.
Yes, yeah. And even the rinse and wash machines for the resin printers, they're actually, you don't need to do much. You understand what I mean?
For like SLA printers where it's
washers, yeah. They have platforms and they have a nice enclosure.
Yeah.
And all you might need to add is a small $2 thermo controller. That's done project. It's very simple in my view. I don't know how others view it.
I guess, I mean, the only thing with SLA 3D printers is usually the print space is quite small. I don't know what the size of what we're talking about is exactly, but it's often a handful of inches or centimeters, really. That's the only thing that I would think.
The recent Anycube one is 20 centimeter by 20 centimeter in width and in width wise, area wise, but the height is little larger. Maybe it might not be super size was not so great, but it's good platform, I think, if one would like to have a desktop incubator.
I got myself an SLA printer last year. I haven't used it much, so I mean, it'd be an interesting thing for me to look at personally as well. So I might have a look into that. But you can put together bigger parts with it, but the print size is just, that's the only thing that I would think, is that for most of them it's quite small. But yeah, if that's big enough, that's big enough,
Sorry, I mean to say the wash and cure system, not just the printer. So wash and cure system also looks like a printer because it has the same area.
Yeah, I got both, because it's handling the resin is just I don't want to be dealing with more health and safety stuff for my for my personal health and safety than is necessary. So it's just kind of like, yeah, that's a cheap thing I can also get alongside it, and I'll just sort it out, if you know what I mean. So yeah, I've got one of those as well.
Great.
I think we're near the end of our allotted time. Bamsi, are you the nominated host this evening?
Yeah, I'm just here. I'm just keeping a type of that. Right. If anybody else has any other questions, you can put that in the chat, or we still have the Mattermost chat and the Google groups there. And thank you very much, Nico. I think Nico left us, but that was a very good session, and we'll see you all again on the 14th of June.
Awesome. Thanks, Bamsi. Thanks, everyone. Really good to see you.
Thank you, everybody.
Thank you all. See you.
Bye.
Bye-bye.
See you soon.
Bye.